G-protein linked receptors are transmembrane receptors that are activated by the binding of a signal molecule. The binding of this ligand leads to a conformational change in the G-protein linked receptor. The activation of this receptor in turn activates and interacts with the G-protein. The alpha subunit of the G-protein will exchange GDP for GTP with the addition of a phosphate. The alpha subunit bound with the GTP will activate a membrane-bound enzyme, in this case adenylyl cyclase. By binding with the adenylyl cyclase, it leads to the creation of second messengers, which will prompt a cell response. This message, originated from the G-protein receptors, may go into the nucleus, where genetic information will be transcribed in order to create proteins and respond to stimuli.
Receptor tyrosine kinases play a role in cell-to-cell communication by receiving signals from its environment. Ligands bind to the extracellular region of two receptor tyrosine kinases that are near each other. The two receptors come together, forming a dimer. ATP acts as a phosphate donor when the phosphate group is transferred as ATP is converted into ADP.
The receptors attach phosphates to tyrosine. The paired receptors phosphorylate each other. This is done through transphosphorylation as the tyrosine from each side acts as a substrate for the other. The now phosphorylated dimer is considered activated once the 6 phosphates are bound to it. Relay proteins will move towards the receptor and bind to the phosphate found on the tyrosine kinase receptor. This will cause a conformational change in the relay protein. When the two relay proteins experience this conformational change, it will trigger a different cell response. Depending on the signal molecule, the proteins cause different responses, the most common being the maintenance of metabolic functions, such as the release of insulin to lower blood sugar, and growth-related functions, such as the release of growth hormones.
Ligand-gated ion channels are ion channels that can open in response to the binding of a ligand. When the ligand attaches to the extracellular region of the channel, the protein structure opens, allowing for ions to cross without having to touch the hydrophobic core of that phospholipid bilayer. When the ligand detaches from the channel closes and stops the movement of ions into the cell. The movement of ions within the cell changes the membrane potential, enabling changes in electrical activity, which is important for neuron signaling in the nervous system.